Good. All right. So, so this is intended to be a tutorial talk, and so what I'm going to be talking about today is going from genetic data to understand the evolutionary processes that might give rise to such data. And so we've already had a few talks talking about this kind of a problem. So Sohini had a great talk on understanding deep human history from genome sequences. And so the setting that we are looking at is we have genome sequences, possibly from multiple populations, say human populations, and we'd like to take these genome sequences and infer the underlying process that gave rise to these sequences. So for example, what might be the relationships between these populations when they split off from each other and split off from other related populations. So, so just to motivate why this problem is interesting, so of course, estimating these population genetic parameters like mutation rates, recombination rates, and population sizes and relationships, those are fundamentally interesting in population genomics. We are interested in understanding why mutation rates are the way they are, why recombination rates differ amongst populations. But also these problems are interesting in a lot of downstream analyses. So when we are looking at association studies and we want to correct for confounding, we'd like to be able to understand the relationships between different individuals in these data sets. If we know the relationships amongst populations, such as in admixed populations, that enables new kinds of study designs, like admixture mapping, which takes into account the peculiar structures of these admixed populations. And when we're doing studies like scans for selection, we need appropriate null models. And again, these kinds of analyses furnish us with the appropriate null model in a lot of downstream analyses. So, so those are kind of the reasons why these evolutionary models are interesting in population genomics. But another thing I want to do in this talk is to use these evolutionary models to talk about inferential problems. And the kinds of inferential problems I'll talk about is one is of modeling, and I'll touch upon one modeling framework, which is graphical models. And a second thing I'll talk about is a framework for doing efficient inference, variational inference. And essentially, these inferential problems are very general. They have nothing to do, they're not exclusively applicable only to evolution or to genomic data. So even if you're interested in neither evolution nor genomics, hopefully some of these things will be interesting to you. All right. So again, just to motivate why we care about understanding evolutionary processes, here is a very simple example where you're running a genome-wide association study. You have individuals who have disease and normal individuals. And here is a SNP which appears to be associated with the disease status. But of course, in this setting, you might have people with the disease drawn from a completely different population than people who don't have the disease. And so this is a situation where the underlying population structure can confound scans for association. And so we'd like to be able to infer this. This is not known a priori, and we'd like to be able to infer and correct for this structure. So here's another example where this was a type 2 diabetes scan that was done in Mexican American populations, and that this study came up with a novel risk variant that's found in this gene called SLC16A11. And so this is a risk variant that confers increased risk for type 2 diabetes in Mexican Americans. And if you look at the geographic distribution of this risk variant, it's found to be at appreciable frequencies in the Americas, very low frequencies in the rest of Eurasia, and essentially absent in sub-Saharan Africa. And so given a pattern that looks like this, we'd like to be able to say, is this unusual? Has this variant been under selection in the Americas? And to be able to do this, we need to understand what is the expected distribution of these allele frequencies. So, so my talk has, I guess, 3 parts, and I'll see if I can cover all 3 of them depending on how much time we have. So the first part I'd like to focus on are the underlying evolutionary processes that form the building blocks in a lot of these probabilistic models. And so there are two broad perspectives in building these models. One is what we call a forward in time perspective, and there's a related perspective which is the backward in time perspective, and both have their own complementary strengths. And then in the second part, I'll talk a little bit about admixture models, and in the final part, if I have time, I'll talk about some applied work that uses these ideas in testing for admixture. All right, so the basic idea is we'd like to first start with a simple kind of mechanistic model for how a population evolves. And one of the very common models that's used primarily because of its simplicity is the discrete time Wright-Fisher model. So the setting is fairly straightforward. We have populations that are evolving in discrete, non-overlapping generations. We're assuming that things are neutral, so what allele you carry doesn't influence how many offspring you have. Mating is random, and for now we assume that population size is constant, and just for convention we assume that there are 2n haploid individuals. So this is what the one time slice of the Wright-Fisher model looks like. So we have individuals, and we've colored them according to the allele they carry. We're going to denote the two alleles that they carry as a 0 and a 1. And these individuals give rise to their offspring in the next generation. And the way each person in the next generation chooses their parents is by sampling independently with replacement. Right? So here is a case where you have 2 copies of the black allele in one generation, and in the next generation we have 3 copies of the black allele. So what we'd like to be able to describe is how the number of copies of the black allele changes with time. Right? And in this very simple setting, the number of copies of the black allele, which is x sub t, is going to be binomially distributed with the frequency parameter depending on the frequency in the previous generation. So that's easy to see. And in this sampling scheme, because it's binomial, what that means is the number of copies of this allele in expectation doesn't change over time, so it stays a constant. And the variance of the number of copies of this allele it depends on the allele frequency, but it also depends on the population size. So smaller the population size, larger the variance. Right? So this Wright-Fisher model is a Markov process. What that means is once I tell you what the state of this process is at a given time, the state at the next generation is independent of the state in the previous generation. And given this setting, we can now ask questions like, if I were to start this process in a given state, what is the state at a future point in time? Right? So those are the kinds of basic questions we are interested in trying to build this model. So it turns out that this is a simple model, but it's actually not so convenient to do interesting computations in many cases. And so often what we do is we work in a continuous time setting. So the idea is we look at time as not discrete generations, but as continuous. And we take the underlying discrete time Wright-Fisher model, and we consider a rescaled version of this model. So we look at x as a function of time, where time is rescaled in terms of the number of generations. So in this new process, 1 unit of time corresponds to 2n units of in terms of generations. And what has been shown in the literature is as the population size becomes large, this rescale process converges to another stochastic process, which is called the Wright-Fisher diffusion. So the Wright-Fisher diffusion is a continuous time, continuous space process. And you can actually write down the transition function for this process, which describes how it evolves. So the transition function tells you if you were to start this diffusion at a given allele frequency x, what is the allele frequency going to be like at some later point in time? So we have explicit analytical forms for this transition function. This was initially computed by Kimura in the 50s. And of course, if now you can also compute the expectation and the variance of the allele frequency as a function of time. And here what we see is the expectation is unchanged with time. So this is analogous to the discrete time case. And again, the variance also has the same dependence on x, and it has a dependence on the time which decays, which actually increases as tau goes to infinity. Okay. So when the drift is small, the variance can now be approximated by a linear function in tau. So this becomes exactly tau. And people often use a normal approximation where they say that y at time t has a normal distribution with the mean and the variance matching the mean and the variance of the Wright-Fisher diffusion. Right? So this often works in practice when you're dealing with populations as in humans, where we know that drifts have been quite small through the history of the human populations. All right, so this is going to be the basic building block. So this is describing what happens when a single population evolves forward in time. And now we're going to take this and try to build on it and introduce structure into the population. So here's how we're going to represent this. So in this setting, we now have a population that starts here, and x sub 1 describes the allele frequency at a given SNP in this population. And this population then evolves for a certain period of time, and that becomes x sub 2. And then x sub 2 then splits into two populations, 3 and 4, which further diverge. Right? So we have a single population that evolves for a while and then splits into two populations. And now we'd like to do the same thing. We'd like to ask, what's the probability distribution of the allele in these populations, given that you've started in a configuration in the ancestral population? Okay. So this notation, if it's not familiar to most of you, we are using what is called a directed graphical model to represent this structure. So every vertex here is a random variable. It tells you what's the allele frequency at that point in that population. And the edges tell you what are the dependencies amongst these random variables. So what we're seeing here is x1 evolves to form x2, and then x2 splits into two populations, and they evolve independently. So what this model is telling us is, once I've given you information about the allele frequency x sub 2, the allele frequencies in x3 and x4 are independent, conditioned on x sub 2. Right? So what we can now do is we can write the joint distribution of the allele frequencies in these four populations. And because of the way this model is set up, and because of the conditional independence between x sub 3 and x sub 4, you'll find that this conditional independence manifests itself in this probability factoring itself. So if you look at the factorization of this probability, you'll have x sub 4 only depending on x2, and x sub 3 only depending on x2, and x2 depending on x1, and x1 is at the root of this model, and so it has no further dependencies. So essentially, you can factorize this model in a way that describes how these populations are related. So this is the joint distribution amongst these populations at a single position in the genome. Now, in practice, what we have are observations from multiple SNPs along the genome. So in this case, now we have observations from M SNPs, and we are observing only the populations at the leaves of this tree. So we have observations only from the populations x3 and x4. So these are present-day populations. And in fact, what we get to observe are not x3 and x4 itself, but samples from this population. So the shaded circles here represent our observations under this model. So what we're observing are noisy versions of these allele frequencies in these two populations at M SNPs. And we're going to be assuming here that these M SNPs are evolving independently. Right? So again, we can write down the joint distributions of all of these random variables. And so this is the joint distribution of the x's and the y's given the parameters of this model, which are the taus. So the taus are the drifts along each of these populations, and they are shared amongst all of the SNPs. So if you look at this joint probability, because we are assuming that these SNPs are evolving independently, it factorizes into m distinct terms. And each of these distinct terms is essentially the term that we wrote down previously. Right? So you're going to have y4, which is the observed allele frequency only depending on x4, and y3 depending on x3. And x3 and x4 are independent of each other given x2. And finally, x2 depends on x1, and x1 doesn't depend on any other variables. Right? So this is the decomposition of the joint distribution. So the goal is we are now observing samples from two populations at these m SNPs. And given the data from these two populations at m SNPs, we'd like to infer the drift parameters. So we'd like to infer the parameters tau 2, 3, and 4 given data from two populations at m SNPs. So the key ingredient to be able to do that is to write down the likelihood. So we'd like to write down the likelihood of these shaded circles and as a function of the parameters that we care about. So this is basically the likelihood is basically the probability of the y variables given the parameters of the model. And again, this of course decomposes into m separate terms because of independence. And each of these m terms is obtained by integrating out the values of the hidden variables, the x's. So you have the product of m separate likelihoods, where each of these likelihoods is obtained by computing these integrals. So now we can look at how do we compute each of these integrals in turn. And the difficulty here is this is a multi-dimensional integral. So essentially, you'll have to integrate out x3, x4, x2, and x1 jointly. Right? And as you can imagine, as the number of populations you're observing increases, the dimensionality of this integral increases, and it becomes computationally expensive to compute these integrals. So here is where we can use the structure of this probabilistic graphical model, or the independence assumptions that we make about how these random variables are related, to compute these quantities efficiently. So the basic ingredient here is something called the sum product algorithm. You can think of it as a way of querying this graphical model to get at subsets of the quantities that you care about. So you have this graphical model which is defined as a joint distribution over this large collection of variables, but you actually care about a subset of them. And so the sum product algorithm gives you a principled way of querying the probability of these subsets of random variables. So just historically, this was these kinds of algorithms arose in the phylogenetics community. So Felsenstein proposed an algorithm for computing these quantities in the context of phylogenetic trees, and they were called the pruning algorithm. There are similar parallel algorithms that some of you might have encountered in the context of hidden Markov models. So we have what are called the forward-backward algorithms. But these are all special cases of the sum product algorithm. And so it gives a unifying way of thinking about inference over trees and chains. So let's look at what the basic idea of the sum product algorithm is. It's basically intuition. It says you have these independencies, so how do I compute these integrals in a smart way instead of having to do this multidimensional computation? So here is the likelihood that you care about. So this is the likelihood for a single SNP as a function of the parameters obtained by integrating out all of the x's, which are the hidden variables. And the basic idea is if you write out this quantity, you can do the computation so that some of these computations can happen before the others. So it's a way of rearranging the computation. So here is one example. So if you look at this likelihood, you have x4 and x4 appearing in two terms. So x4 appears with y4 here, and it appears with x2 here. And those are the only two terms in which it appears. So you'd like to do that computation first. Similarly, x3 appears in these two terms. You do that computation first. And of course, these two computations are going to be a function of x2. So you take these two quantities, you couple it with the other term that depends on x2, which is x2 given x1, and you do that next. That's going to depend on x1, and you have another term that also depends on x1, so you pair it with that, and you do your final integral. So by rearranging the computation, you're able to do this more efficiently. And so the way to write this is you can think of it as computing these partial integrals. So the first integral is got by integrating out, so I think there's a typo here. You're integrating out x4, and you're getting a partial integral that depends on x2. You're integrating out x3 and getting another term that depends on x2. Then you're taking these two terms and combining it with the other term that depends on x2, integrating that out, and you get something that depends on x1. And finally, you take x1, you take this quantity, combine it with x1, and integrate it to compute your final likelihood. Okay. So this turns out to be much more tractable, and it allows you to compute the likelihood fairly efficiently. So that's the general idea. So now that we have the likelihood, you can do the other kinds of inference problems that you care about. So with this likelihood, you can now ask what is the maximum, what is the value of these drift parameters and the tree topologies that maximize your likelihood. Or if you're in a Bayesian setting, you can look for computing the posterior probability over these parameters. So there are a lot of methods to do this, and there's a rich literature to do this in the phylogenetic setting. And here we have not made too many assumptions about the evolutionary models themselves, but in practice people have developed further approximations. For example, if the drift is small, you can make approximations about normality, and those can enable faster computations of these likelihoods. Right. So this was about a simple case where you have a population that's structured as a tree. There are lots of other extensions that you can now consider. The first is admixture. So admixture, one of the reasons to consider it is it's something that we've realized is really common in human populations. There are lots of major population groups that are admixtures of other population groups. And so we'd like to be able to build models that actually take admixture into account. So simple tree-like models don't seem to be a good fit for a lot of human populations. So that again is something that you can add into this model. So here is this model with a new population, population 5, which is obtained as an admixture of populations 3 and 4. And from the point of view of the evolutionary process, the allele frequency at 5 is obtained by basically taking the frequencies at 3 and 4 and combining them according to the admixture proportion alpha 1 minus alpha. So this is people have been looking at these kinds of models more recently. Things turn out to be much more complicated when you have admixture admixtures appearing. So now you've gone from a tree to what's called a polytree, and what that means is this increases the computational complexity of the likelihood computation. All right. So now I've talked a little bit about the forward time process. Now I'm going to switch gears and talk about a different perspective, which is the backward in time process. Okay. So here again, we are going to start with the Wright-Fisher diffusion, with the Wright-Fisher the Wright-Fisher model. And so we have individuals who are present today, their ancestors in the previous generation, and so on. And what we're going to be doing is we're going to be looking at a subset of the individuals and tracing their lineage back in time. So here we start with 3 individuals, and we trace their lineage back in time. At some point of time, these individuals might find a common ancestor. So in this case, the 2 ancestors of these 2 individuals find a common ancestor at this generation, whereas these 2 individuals find a common ancestor at this generation. So we can ask, what does the ancestral relationships between the sample set of individuals look like? And that is, of course, represented by this tree. And we can also say something more. We can say that this ancestral process is also a Markov process. So if I tell you what are the sets of individuals existing at a given point in time, their ancestors at the next point do not depend on the individuals at the previous point. So we can represent this by a quantity a sub nt, which tells you how many individuals exist at every point in time. So you started out with 3 individuals. In the 0th generation, 3 individuals in the first. Then there's a coalescence. You have 2 individuals who then coalesce. You have 1 individual, and then the process stops. So this is a process for which we can write down the transition density. And it turns out we're going to do the same trick as we did in the case of the Wright-Fisher diffusion. We're going to look at what happens to this process as as your population size n becomes large. So you're going to have 2 quantities, the population size uppercase n and the sample size lowercase n. And we can write down what this transition density looks like as the uppercase n quantity becomes large. And the basic idea is this transition density, it transitions between having I individuals in the next generation given I in the previous generation. You can have a coalescence which reduces the number of individuals by 1. And of course, you can have multiple coalescence that reduces the number of individuals by more than 1. And the basic idea is as the population size becomes very large, the chances of having multiple coalescences decreases. And so you either stay in the same state or you decrease by 1. Right? And so you can ask, what's the probability that you'll stay in the same state for a given point in time? And that turns out to be geometric with this parameter. And as n becomes large, this converges to an exponential distribution with a rate parameter that is given by the number of extant lineages. So the coalescence is essentially this limiting ancestral process. You're starting with some number of individuals at your leaves, and each of these individuals then coalesces at different points in time. And so what you get at the end of the day is this tree, which is a rooted binary tree, where each of the coalescence points is annotated with the times when these coalescences occur. And the time to the next coalescence is given by an exponential variable whose parameter depends on the number of lineages that exist. Right. And of course, every pair of lineages is equally likely to coalesce because we are assuming neutrality and random mating. So this is under a constant population size. We can also ask what happens when the effective population size changes. And changing the effective population size changes the rate of coalescence. So if imagine a case where the effective population size changes to 1 individual, then everybody is going to coalesce, and so you're going to see a lot of coalescences in that point in time. So what we can do is we can turn this around and use the coalescence rate to infer the effective population sizes. And so Sohini's talk went into a lot of detail about how you can use the coalescent process to learn about effective population sizes over the history of a population. So just to summarize, so you can think of these forward in time and backward in time models as 2 complementary ways of modeling the evolution of a population. In fact, it turns out that they are mathematically equivalent in some sense. And I've briefly talked about extensions to the case where there is admixture. That's still a fairly complicated situation. We know a lot less about how to handle them compared to the case of simple phylogenetic trees. Other kinds of extensions that that people have been working on are how do you deal with geographical as well as temporal information. For example, what you might get out of ancient DNA. Recombination is one of the complicating factors here. It's always easy when you have independent sites. And again, selection is also quite challenging to handle in these kinds of models. All right. So now I want to talk about another way that people have been looking at admixture, which is not looking at it from a detailed evolutionary perspective. But instead, the model is cruder, but aims to get at other kinds of information. So to understand this, we'll have to dig into a little bit of what these admixture processes look like. So here is a situation where we have two populations, blue and red. They're admixing, so they give rise to offspring, who in turn give rise to further offspring. And the key process that we are interested in here is now recombination. So here what happens is, if you look at the offspring of these two individuals, this offspring inherits an entire chromosome that's blue or red. But when this individual transmits down to the offspring, because of recombination, you have a chromosome that's partially red and partially blue. Right? So recombination essentially shuffles up these chromosomes as they get transmitted in every generation. And if you look at the present day set of admixed individuals, they essentially have this patchwork structure, where some portions of their genomes come from blue and others come from the red population. So this quantity, this ancestry along an individual's genome that can potentially change as you look at different individuals is what's called local ancestry. It's local to a specific region of the genome. And we have another quantity which we're interested in, the genomic ancestry, which is the average proportion of the ancestry that an individual inherits from distinct populations. So there's been a lot of interest in trying to build models that allow us to infer genomic as well as local ancestries, especially given genomes from the ancestral populations. So here is one model that has been that has been one of the mainstays of admixture modeling. So to quickly describe what's going on here, so we have observations which are genotypes. So we are observing at I individuals at J SNPs, their alleles. And each individual can have ancestries from multiple different groups. And the way we think of this is at a given SNP, an individual can have ancestries drawn from any of the ancestral populations. So that's given by this variable Zij. And conditioned on the value of this variable Zij, the individual draws the alleles from the allele frequencies for one of k populations. So basically you have k populations, and depending on what is the ancestry you have at a given SNP, you draw your alleles according to the allele frequencies in that population. Now finally, of course, each individual has their own admixture proportion. So that tells you what proportion of your genome comes from the two different populations. And you draw the local ancestries at each SNP according to the admixture proportion that you carry along your genome. And finally, in a fully Bayesian setup, these parameters have additional hyperparameters that allow you to share information across these variables. So given this model, the goal of a lot of admixture inference is to infer the hidden quantities. So you like to infer the local ancestries, the genome-wide admixture proportions, and the allele frequencies in the ancestral populations given genotype beta. So we are interested in computing this posterior over the hidden variables given data. So a traditional way of doing this, in general, this posterior is intractable to compute. And so one approach that has been widely adopted is to do Markov chain Monte Carlo. So the idea is you approximate this posterior by drawing samples from a Markov chain, and you set up this Markov chain so that its stationary distribution is the posterior distribution that you care about. And there have been several proposals to do this, and one particular proposal that works well in this class of models is what's called Gibbs sampling, where you can basically compute the conditional probability of each of the hidden variables given all the other hidden variables. So a challenge, of course, with these MCMC methods is it is actually quite difficult to get them to scale on large data sets. Convergence to the true posterior can be a problem. And so people have also been looking at deterministic approximate inference methods. And one promising class of methods is what's called variational inference. So a simple way of summarizing variational inference, in the words of David Bly, who's done work on topic models, which are a related class of models, is it's the algorithm that you implement as you wait for Gibbs sampling to converge. So the basic idea of variational inference is you convert this problem from a probabilistic inference problem to an optimization problem. So you have a certain objective. We can talk about what the objective really means. And this objective is a function of a distribution. And so you try to find a distribution that maximizes this objective. And so what is this mysterious objective? So this mysterious objective is looking at, it's a function of this distribution that you're choosing. And you're computing the expectation of the posterior relative to this distribution, as well as an entropy term. And when you work out the math, it turns out that this objective is a lower bound on the true likelihood that you'd like to optimize. So by maximizing this objective, you're maximizing a lower bound on the true marginal likelihood. And it turns out that another way of looking at it is it's equivalent to minimizing the KL divergence of this function to the true posterior. So you're looking for a distribution that minimizes the KL divergence. The KL divergence is a way of measuring divergence between two distributions, and it's 0 only if the two distributions are identical. So of course, to minimize this, you will set K exactly equal to the posterior, but that kind of defeats the purpose because that's a quantity that you cannot access. So instead, you restrict Q to a smaller class of distributions that are tractable. And so that's where the design decisions come in here. And there are many ways of setting up. Yeah.
How does that change the Kullback level? Because that's the difference between entropy of Kullback level is KL divergence is.
KL divergence is essentially the integral with respect to Q of log Q over P.
But there, the above term is entropy over P minus entropy over P. So how does that become?
So the expectation is always relative to Q here, right? So the expectation is relative to Q. So that's something that I should have made clearer. Yeah, you're right. There is a whole class of methods which does expectation relative to P. And that leads to different properties. So there are expectation propagation methods that do expectation relative to P. All right. So one way to search for a tractable class of distributions is to assume that your distribution factorizes in a simple way. So you have a bunch of variables, and you have a product distribution over these variables. And this leads to what's called a mean field variational inference, or a mean field approximation. And once you make this assumption, it turns out that solving that problem can be done by doing coordinate descent on each of these terms in turn. So you can work out what these updates look like. And so essentially, the form of this algorithm is going to be something like this. You're going to be looking at your objective, and as long as your objective is changing, you take each of your terms and you update them by computing the expectation of the posterior relative to all the other terms. And because this is factorizing, these expectations can actually be computed efficiently. And you do this till it converges, and finally you return this factorized distribution as your estimate of the posterior. It turns out that in this case, in the admixture model case, things are even simpler. Although we have made no assumptions about what these distributions are, they actually turn out to have simple and analytic expressions. So each of these will have, the class they belong to will be either multinomial, Dirichlet, or beta, because that's the conjugate distribution in this model. And so essentially what you're going to be doing is in every iteration, you're going to be updating the parameters of each of these distributions. And so finally it boils down to doing this, where these are the parameters associated with each of your distributions, and you're updating them. And the thing to realize is these are coupled. So when you make an update to one set of parameters, that propagates to the next, and you keep doing this till you converge. So people have implemented variational inference in the context of admixture models. So here is an example from this paper by Raj et al., which does one kind of variational implementation. One thing to keep in mind is I've just talked about the very basic way of doing this. In practice, there are lots of additional tricks that can be done to speed up convergence, and that's something that they explore in this paper. So just to give you the highlight, they looked at a demographic model with 3 populations that admix, and the key quantity they're looking at is a measure of how closely can you approximate the genome-wide ancestry proportions. When you look at fast structure, which is this variational inference for the admixture model, and they compare to admixture, which is performing EM inference on these admixture models. And they find that they get competitive results, so it's promising. And they do find that computationally, it's a little more expensive than doing just EM because they're running a fully Bayesian model, but it's much more computationally efficient than doing classical structure, which is doing GIP sampling. So they can get competitive results while still being computationally efficient. They also talk about some other results, but I don't want to spend too much time on this. So just to summarize, this separate class of models, these admixture models, they are less detailed than the kinds of evolutionary models that we were looking at, but they're more tractable in general. So you can do things like local ancestry inference, which would be much harder to do in an evolutionary setting. And I've talked about one way of doing computationally efficient inference using variational algorithms. It turns out that even variational algorithms can be very slow for really big data sets. People have now been looking at alternatives to this, and that's still cutting edge research. Of course, there's no such thing as free lunch. Just because we have a computational and tractable alternative doesn't mean that we have actually solved the original problem. So although these variational algorithms tend to give you good point estimates, it turns out that they underestimate the standard errors. So you tend to be more confident of your parameter estimates than you should be. Another thing that's lagging behind is for MCMC, there's a rich body of theoretical literature that kind of tells you when and how it works. There's less of an understanding of this for variational inference, and it's still very much active research. So I have one more section, but I think I'll save that for one of my later talks. I think I'm out of time, and I can take any questions.